Right. So look, I'm really keen to get started. And yeah, good morning, afternoon, or evening, depending where you are in the world. But first of all, thank you so much for joining and welcome to the exciting event for BioTalent Social. So this is our homegrown initiative. The idea is to connect with our networks by discussing the relevant topics that impact our industry. So what we've put together is a panel of experts from the cell and gene therapy sector. So the panelists all play key roles in development and commercialization of these life-changing therapies. They're all well versed in what it takes to ensure that new innovations reach their potential, but also what are the next areas for development. It's very clear the pace of development within the sector is increasingly accelerated, with the challenges faced by companies, by startups, startups and large multinationals. They're so diverse and range from the securing of funding, how to scale up, regulatory challenges, and of course, finding the right people with the right skill sets to grow their business. But from my perspective, what is inspiring is that there is so much potential to be realized within this sector, but also I'm very conscious to promote, because as a sector, I feel that we don't always promote ourselves effectively because the capabilities and the impact these therapies are having and will continue to have, with a number of success stories still to come through, new breakthroughs and successful results of clinical trials are, all aspects that are definitely worth celebrating. Now, the idea of tonight's session, is to hear from experienced professionals who are right at the forefront of the sector, and of course, whose advice will be very helpful for businesses as they navigate the path to success. So what can you expect? First of all, there'll be about 45 minutes of a panel discussion with a 15-minute Q&A at the end of the session. After this, there'll be a half an hour networking for those who wish to continue the conversation further. So, first of all, introduce myself. I'm James Hume. I'll be the host and chair for the webinar this evening. And to help add some context, I've been recruiting in the cell and gene therapy sector for nearly 10 years now. So from when the sector was tiny to where it is now, and I'm excited to continue to be a part of this innovative and fast developing sector. My area of focus is on the R&D and translational sciences. So that's everything from discovery through translation, scale up to GMP manufacture. Look, 10 years is a long time to be any sector within recruitment. But for me, why I've stayed in for so long is that science, the pace of development. I find it fascinating, but it's the impact these therapies have on people in society. For me, I always find that very engaging. And we want to make this discussion engaging as possible. So if you have any questions for our panel throughout the webinar, please feel free to ask them in the side panel or in the Q&A section at the end. We'll be able to get through as many as possible. Can't promise we'll get through them all. We'll do as best as we can. So before we kick things off, please note that this is being recorded and will be uploaded to our BioTalent Social YouTube channel, where you can also access our upcoming webinars and podcasts. So without further delay, I'd like to introduce a fantastic panel of 3 leaders within the sector, all who impact in a significant way. So I've done a lot of speaking already, so I'd like to hand over the panel, beginning with Stephen. Yeah, could you just take it in turn to introduce yourselves, your role, and just, get a bit of sense of who you are.
Hi, my name is Stephen Sullivan. I'm a consultant for Linval Bio, so we consult on a range of different items pertinent to pluripotent stem cell therapeutic development and induced pluripotent stem cell development.
Cool, thanks, Stephen. And
Shall
Kerstin?
I can go next.
Yeah.
My name is Kerstin Papenfuss, and I head up the pharma sector at Deep Science Ventures. Deep Science Ventures are a venture creator, and what we do is we create novel biotech ventures or deep tech ventures from scratch, focused with the outcome in mind. Before joining DSV about 2.5 years ago, I was leading commercialization of research for the cell and gene therapy catapult, and been working at the kind of forefront of getting ideas out of the academic realm into the pharma sector for the last 10 years or so. John, over to you.
Hi, everybody. Good afternoon. So I'm Dr. John Council, so I'm a lecturer at UCL. I've been based at UCL for about 10 years. I have a research group here, and we develop new gene therapy technologies. So we try to look at what goes on in the inner working of gene therapies, how we can engineer them to make them better. And specifically what I do at UCL is I work with clinicians to see if we can get those new technologies into translational research pathways so they can actually turn into products and hopefully help patients.
Fantastic, amazing. Well, right, let's get started with the first question. So Cambridge University, 2019, has previously described technology transfer offices, or TTOs, as playing a critical part in turning early stage inventions into products or services that benefit everyone. So what I'm keen to understand is, how much are we currently doing with you in the UK regarding TTOs and the commitment to benefiting the consumer when creating CDT products? I guess, Stephen, would you like to start?
Yeah, I mean, the universities are in a particularly difficult situation, I think, at the moment, with so much disruption that has come from situations like COVID, and that has hit things. So there's sources of revenues that universities could normally bank on that are now kind of disrupted. So, I mean, one of the key things that we hope is that the tech transfer offices don't seek to generate additional revenue through tech transfer that would ultimately actually arrest the progress of new therapeutics to the clinic. So, it's quite important to be pragmatic, especially at the early stages of developing new therapeutics, that, everybody realizes that therapeutic development takes time. It takes a lot of different specialities all working together in a synchronized way. And, looking for too much too soon could actually cripple your business that's coming out of the university. So, I mean, and one of the things that, I tend to look at things from a global perspective. If you look at, the States and Canada in particular, that there's very good pragmatism in the universities that facilitate companies cutting deals that won't impair them early on. So, I think that the UK has got a lot of really good, strong pieces of infrastructure. So you've got the NHS, which is integrated healthcare, provision, and then you've got really good government-funded initiatives like the Cell and Gene Therapy Catapult, which seeks to de-risk key elements of the translational journey. And it would be a shame if tech transfer people don't allow the kind of creative thinking to allow companies develop and prosper, and that they don't kind of look for quick money at the expense of the long-term stability of the company.
Yeah, no, I think, well, thanks for that. I mean, yeah, totally agree. You want to give the companies the best chance to get their therapies to the right people. That's so important. And the TTOs definitely have a key part in that. I guess, John, obviously being in your position, I mean, what's your take on the role TTOs play and how they can best facilitate?
So obviously my experience is quite limited to UCL. Obviously, I would say biased, but I'd say UCL do quite a good job of ensuring that things move forwards. UCL have got quite a good track record in terms of commercialization of gene and cell therapies. I think that's partly due to the fact that there's so many hospitals affiliated with the research groups that really helps to accelerate things. We've got a very good working relationship with the clinicians. The clinical and research teams are well tethered together. So if I want to discuss a potential idea with a clinician, I sometimes don't have to knock on their door or email them. I can just walk in and start talking. And that's, I think, where UCL comes into its strengths. In terms of the tech transfer offices, it's a similar situation, actually. We work very closely together. And I think that really helps move things forwards. In terms of how things then progress at the commercialization end, in terms of, I think what we're kind of getting towards here is more like who takes a cut of what size is that cut. how is that going to affect the attractiveness of this? Yes, I hear different things about that. I'd say practically the relationships with the tech transfer offices are very good in UCL and in the UK. In terms of the rest of it, I'm not too sure myself what the significance of the current relationships are, but certainly I'd like to have a bigger cut if possible, but that's pretty obvious, isn't it, really?
Good stuff, but it's true. You can get a bit bogged down in the cuts and bits like that. I imagine that's a bit complicated. But Kerstin, from your experience,
yeah, so from my perspective, I've been working with tech transfer offices obviously for the more than 10 years by now, and been in a position where I can actually compare what's happening in the small molecule and antibody space and the cell and gene therapy space, which I've been doing much more of late, right? And what I can see is obviously tech transfer offices are key to the translational process, and they are great. But for the cell and gene therapy space specifically, some are underfunded or under-resourced in terms of access to talent. So I think John is very lucky with his UCLB example because they are fantastic. But I have been working with other TTOs that just don't have the necessary understanding of what it takes to commercialize a cell and gene therapy. And that's when they end up providing the wrong advice to the potential academic founders, just because they haven't seen that kind of thing before. Or they are putting people on the board that don't necessarily have the expertise to help these either. So I've been dealing with all of this. As well as then looking at this kind of commercial attractiveness piece that Stephen has said. So it takes a lot more money to actually get cell and gene therapies to a value inflection point as compared to small molecules and antibodies. And therefore, I think universities should be setting these companies up with a smaller share to start with, to set them up for success rather than failure, because the entrepreneurs who are taking these forward need to be ultimately motivated to benefit as well.
Yeah, it is challenging because, of course, a lot of these are very new, novel, cutting edge. So there is that lack of knowledge, and trying to get that knowledge in the right place can be challenging. But well, with all your good work, hopefully that will certainly be overcome. But thinking a bit more bigger picture, I think so often from my perspective hear a lot about investment coming to this sector, whether it's from bodies already within or those from outside. It's the bit from outside I'm thinking about. What is it, in your opinion, that makes the cell and gene therapy sector so attractive to invest in? I think, Kerstin, it'd be good to get your perspective on that.
Yeah, I think that's solely to do with the potential for cures that we see in the cell and gene therapy sector. So if you get a cell or a gene therapy to work that has the potential to cure the disease, then you have the potential to disrupt the whole market and potentially get the whole market share, which is not something that you have with a lot of other modalities, which are mostly focused on disease management rather than curative approaches. And I think that's where the whole attraction lies.
Yeah, I mean, for me, I said it a few times in the introduction, the fact that these, broadly speaking, therapies can provide cures, that's just so inspiring, and I think it's just very, very exciting. But Stephen, from your point of view, what does it make?
I mean, if you look at traditional efforts to interrogate human disease and development, they're normally limited by access to material. So, for example, if I'm studying a Parkinson's disease, if I extract neurons from that Parkinsonian patient, they're going to get worse, right? But now with kind of the cell, the development of induced pluripotent stem cells, I've got the capacity to make dopaminergic neurons that can approximate, what happens in the patient outside the patient. And, so you can think about disease modeling, and then you can also think about using those cells as therapeutics. And so the key thing is that, there's a platform of new technologies that are kind of getting around the traditional problems that have beset, researchers and developers and clinical developers in particular. So it's an exciting time, and people can see how, the new technology is getting around old problems. And I think that's, the large part of the the interest in the area, this platform that can generate any cell type in the human body, and how this could be used directly and indirectly for therapeutic benefit.
Yeah, I still find it amazing how you can potentially derive any cell type in the human body, and the implication of that is fantastic. So yeah, more development in that area would be wonderful to see. And then John, from your perspective, what is it about the sector that you see or hear about that makes it so attractive to invest in?
So obviously I'm quite often on the other side, so I'm looking for the investment. So I guess more in terms of like how I see it as being a good sell to people, really, I guess is how I'm going to frame this. But I think historically, a lot of the interest in gene therapy came from those natural, logical situations where you would apply it. So for example, monogenic diseases where disease X is mutated, if you can get a replacement copy of disease X into the right cells, then hey presto, you've cured the patient. So I think the attractiveness there, really, in the first phase of gene therapy was always that really clear target identification. You knew exactly what you wanted from your drug modality. You just need to get that gene into the vector and into the right cells. And it's obviously much, much, much easier said than done. It sounds brilliant on paper, but much, much harder than that to do. But when it works, it's been brilliant. You know, there's some famous examples, particularly the spinal muscular atrophy gene therapy. It's been very, very effective just following that strategy. I guess the next stage of gene therapy, and what a lot of people seem to be interested in now, is how can we then take that beyond the monogenic diseases? How obviously there's lots of other gene therapies that don't target monogenic diseases, for example, CAR-Ts and other ways where we're engineering cells. But for the straightforward gene therapies, how can we take it on to the next step? I would say it's really going for those undruggable targets that people would like to manipulate within a cell, but the drugs don't really manage to do that. So in the cancer field, for example, there's a lot of examples you can come up with. I think with good gene therapy delivery strategies, more targeted gene therapies, we can potentially now go after those, having, I guess, not really cracked the monogenic diseases, but certainly made big advances by going for those clear targets. So yeah, so I think in terms of the history of gene therapy, that was always a selling point. Going forward, I think now we can really motor on and take that momentum into the next phase.
Yeah. I mean, that's truly exciting to hear about that momentum to the next phase. You're moving forward from what already life-changing therapies. And then just changing tack slightly there, John, you mentioned about that delivery side. I think it's interesting with delivery. I mean, I've been hearing a lot more about viral or non-viral delivery, and kind of that seems to be sort of quite a development area. I'm thinking, just focusing on that for a moment. I mean, yeah, is that delivery going to be an area of development? And if so, what are the benefits between, I guess, viral and non-viral from your perspective?
Yeah, I mean, that's always been a big central aspect of gene therapy, particularly looking at things like in vivo gene therapy, but also ex vivo as well. And it's all really come to the forefront even of public discussion, I think, where the lead COVID vaccines that a lot of people had over the last couple of years were based on gene therapies. People started to learn what was a viral vector, what's an mRNA vector delivered in a non-viral liposome. And people became more and more aware of the differences. And obviously there were certain aspects to those vaccines that made some better than others in certain ways. So I think that's been a really interesting effect on the gene and cell therapy space. And certainly a lot of people that I talk to, that they're interested in, how can non-viral vectors be made to work better? What's the possibilities with those? A lot of the successful gene therapies to date have been based on viral vectors. Obviously, the more we can get towards a designer, non-biological type of gene delivery system, the more it could be saleable. So I can see why there's a drive to go towards non-viral vectors, but quite often, until you really start to get to work with the different technologies, you don't completely see the need for using different systems. Historically, the viral vectors have always been used for delivering DNA. Getting DNA into the nucleus of a cell was a really challenging process. The viruses have evolved very cunning techniques to navigate that process and get things across the nuclear pore, for example. So that's historically why they've always been used. I think with the mRNA vectors, perhaps you don't need that, but there are still good aspects to viral vectors that make them suited to that as well. So I'd say all in all, I could compare and contrast these all day, but I think we're at a point where there's so many new technologies developed that depending on what people want to do, you can now pick from that toolkit and just use whatever makes most sense, depending on what it is you're trying to achieve. And I think that's quite a nice position to be in at the minute in the G&CT space.
Yeah, I mean, that's I think you understood that it's a brilliant position to be in. I think that's exciting to see how it can impact future developments. And Kerstin, I know we've also discussed that different delivery methods previously. And what's your take on it? Well, that whole delivery conundrum toolkit from your perspective.
Yeah, I think I'm with John on that one. I think it will be horses for courses, right? There will be indications where your viral delivery mechanism is perfectly suited to the problem that you want to solve. And there you can probably proceed a little bit faster because of all the wealth of safety data and biodistribution data that are already out there, right? For nanoparticles, lipid-based nanoparticles is like a catch-all phrase for very different particles, actually. Just because LNPs work in one context doesn't mean a different LNP would work in the same. So there's a lot of stuff that we still have to figure out, right? And there will, that challenge of getting stuff into the nucleus is very, very key for stable expression. So in contexts of disease where you need to rely on something that needs to be stably expressed for years, then you really need to figure out whether the LNP is the right delivery strategy if you can't crack that nuclear delivery. So it's really horses for courses, and what's the best way? If you want to repeatedly deliver mRNA in a cheaply scalable fashion, then LNP might be the preferred strategy, and that's exactly what we've seen with the COVID vaccine, for example, right?
I see. That helps, I guess, bring it to reality with the COVID vaccine, but also that wealth of choice, and then understanding what the best delivery method. I can see how there's going to be more increasing focus on that area. And then Stephen, what's your point of view? Because I know obviously you're more iPSCs, but it'd be interesting to see what you're.
One of the silver linings to COVID is that people now are used to comparing the price of developing a vaccine or a new therapeutic with the cost of not having it. And that's really, like if there's a huge societal need for a new therapeutic, it's important to measure, the cost of developing new therapeutics versus, the ongoing kind of treatment of the symptoms approach, if you will. So, I mean, I think one of the things I wanted to articulate, and I think this group kind of illustrates it very well, is that cross-functional teams are really important to efficient translation to the clinic. So, early developers in universities, they're making decisions that have huge consequences for later on in the translational journey. And I think one of the things we need to do is to enable the earlier developers get access to other forms of expertise pertinent, required for efficient translation. So this would be areas like, regulatory affairs, understanding what the regulator is looking for with regards to risk and getting approval for clinical trials and, subsequent rolling out of new therapeutics. Manufacturing expertise. There's key decisions that an early developer can make on their own that can completely make impossible scalable manufacturing. And this comes down to the complexity is the key word when it comes to developing new cell and gene therapies, and it really does take a village. So just like illustrated here, like James, you're dealing with the skills and the recruitment issue, which is of huge importance to the field as a whole, and your success or failure will govern just as much as the rest of us the growth rate of this particular field. So it's about us all kind of understanding a little bit of what's going on in each other's professional silo, so that we develop a holistic, vision for efficient translation, and that we can, basically understand how to make life easy for each other. as we, undertake our own particular function in the translational journey to the clinic. So cross-functional teams and, early developers need to be better supported so that they can, make these cross-functional teams that will greatly assist the efficiency of translation to the clinic.
Yeah, Stephen, loads of great points raised there. I think, certainly I feel that, and my colleagues do that, staffing pressure each day, try and rectify that. But and I could talk about that a lot. But yes, that collaborative mindset of everyone working together. So each in that silo, each of their own importance and impact to the sector. Yeah, working collaboratively is the way to bring these therapies to those who need them most. And we've all touched on a couple of the challenges faced. So that could be the knowledge, it could be scale up. They just run and run and run on. But one thing to be a bit more specific is that in 2021, McKinsey reported that more investment in European CGT sector is needed in order for business to thrive. Now, that's not necessarily new news, but I think just to help bring it together is, what do you individually believe are the current challenges regarding investment, startups to scale up, to commercialize? And big question, but how to combat these? Or what do you see as the best way to combat them? I guess, Stephen, you've already started on that. Could you elaborate a bit more?
I think, business people in general are quite pragmatic, and they will always kind of look for the win-win scenario. And I think that sometimes when you're coming out of, let's face it, a resource depleted, academia as it is at the moment, where, academics have to work extremely hard in order to take things through to early stages of development, usually with, limited or unstable resources. So, constantly looking for grants, hoping they come in. But, the key thing is that ultimately, the resources need to be put in place, but so that, you can enable the earlier developer, but also to realize that many other things have to happen after the proof of concept has been shown in order to roll out a new safe and efficacious treatment in the clinic. And that there's just because you mightn't see it on a day-to-day basis doesn't mean it needs to be undertaken. And really, I think that the best way to enable efficient translation is to, really think hard about the resources that early developers need. And this would be like the cross-functional teams, consistent, funding, because, if funding is always erratic, then you can have really good proof of concept, but you're so busy, trying to get the next grant that projects fall by the wayside, and people start following the trends of getting grants rather than, they've developed a really good proof of concept, and can we maintain the focus there to push it through to the next stage of translation?
That proof of concept prove everything, but yes, there's always further challenges always to overcome, and it's always interesting to hear what they are, and also how. I just love solving problems is what we do, right? But Kerstin, what about from your perspective, and particularly what you do at Deep Science Ventures? What do you see as, I guess, such a big question, but the key challenges you face right now and how to overcome them?
I think it all goes back to looking at the problem holistically, and that's what we are trying to do from the outset. So I remember when I was working at the Catapult, I did spot a lot of opportunities where, let's say, Professor X had a good idea for a gene therapy, but didn't necessarily have an understanding of the right delivery strategy and how best to scale this up and what the health economics implication would be. And that always then put me in a really hard position. If that understanding wasn't right, that means that the wrong experiments were done. And you can't really go to venture capital and say, the underlying idea is good. I know we've done the wrong experiments. please give me money to do the right experiments. That's not how venture work and can't work. So I think getting, first of all, education right, so that people understand that they need to look across silo from the get-go, and don't shoot themselves in the foot with early experiments that then preclude things like scale-up, for example. So basically elaborating on the point that Stephen made earlier. But then I think we also need to understand that whatever we're doing, we're not just competing in the UK, we're competing on a global scale, right? And there is still very kind of limited understanding, first of all, in the kind of grant awarding bodies, that for cell and gene therapies, it takes larger cash requirements to get to IND than it would take for a small molecule and an antibody. And you still have to kind of compete with small molecule and antibody programs, and then it's just, okay, this amount of money will get that project to that inflection point, but for cell and gene therapies, that just doesn't work out. The math doesn't work out. So I think having dedicated calls with people who understand that is key. And then on the VC side, also realizing that they just need to write larger checks. And yeah, just maybe then having educated bets rather than having, you know, larger bets. I mean, I always hear that interesting statistic that, you know, out of 100 startups, only 2 or 3 survive. So let's fund 100 to make sure that 1 or 2 survive. But for me, that's like a self-fulfilling prophecy, right? Why don't you put more time into figuring out what's the right one to start with?
Yeah, I mean, that makes it so very clear, because you fund the right ones from the very beginning, you're taking a lot of the risk out, but also you're giving those who've got an opportunity, who have a good idea, good content, good therapy, the platform to actually bring that to the people it needs to, or bring it to the right stages. Yes, it's just a more knowledgeable approach, and that word knowledge has come up quite a few times. I mean, we've all said it a few times already during the webinar, but no, that's brilliant. I love the clarity. And then John, what about from your point of view? I know it's slightly different, but yeah, from your point of view, what those challenges are, and kind of any way you can see to overcome them.
So yeah, the same perspective, really. I think some of the things that I've tried to move on beyond basic research and early translational proof of concept research really doesn't always get tested for feasibility with an academic environment, and specifically from the perspective of things like manufacturing and the regulatory side, just as the things you were mentioning, Stephen. So fortunately, I've been connected with people that do have experience in those areas. The problem is, in the gene and cell therapy space, I would say that the expertise is quite limited. Certainly expertise from people that really get what you're trying to do as well. So again, it comes down to having this like cross-disciplinary understanding within the field, because it's so new that it's not always there. So the people that really do understand the manufacturing side or the regulatory side don't necessarily understand gene therapy. But fortunately, I've been managed to connect with those people. That's been really helpful. And it's made me realize just how expensive it can be to get something all the way to a trial, and just like where the challenges are from that perspective as well. Been quite eye-opening, really. But in an academic environment, you rarely get that, because certainly at UCL, we don't really have big manufacturing groups or people working on that side of research. So I think as time goes on, people become more aware as people are taking things towards clinic. But if there was more of a mechanism to get a pool of people that really understand everything across the board, or had spent time in different disciplines, and that really could be quite powerful, I think.
Yeah, I think one of the points that Kerstin made during our rehearsal conversation, I think is very pertinent. Is it was about that people not be afraid to kind of go into slightly different areas of the translation journey. So if you're an academic scientist, that you're not afraid to go and get some, say, project management or business expertise, or to go and work in, with an investor to understand how they see the world. I think that was a really good point, that experience cross silos can make you really, really useful, I think.
Yeah, over to you, Kerstin, because that was a very powerful point, or is actually.
Yeah, because like, yeah, sorry, is your point, Kerstin?
Oh, yeah, you know, that was the point, that in VC we rarely see operators and vice versa, right? What you see in the US is often that people in VC have been experienced operators in companies and actually built their own companies and then moved into VC, and are also fluidly moving out of VC. And we don't see that as much, right? We see people with a scientific background at the beginning of their career then decide we're going to be in venture, and then they kind of build venture careers. But that then doesn't mean they have the kind of boots on the ground experience of what it really takes to develop a gene therapy, which can then also be really, really hard to take for entrepreneurs that have to face these challenges, that then routinely get told in a board meeting, oh, but this should take you this amount of time. And that can be quite discouraging, right? Whereas if you have like been through that yourself and seen how this works, you have a much better understanding of the actual challenging and how difficult some of these things really are.
Yeah, I mean, definitely. And having the opportunity to work in different silos, cross platforms, have that understanding so that when you're in those board meetings, the right information is passed on, the right decisions made. And you know, that's not just going to happen out of thin air. The knowledge needs to be shared. You need to give the platform to do it. And I think what I'm stumbling over is it's a lot of that's going to be the evolution of the sector, but it's taking up John's point, it's that putting that mechanism in place. I think how to do that, how to get there, and get that commercial experience. I think also in the rehearsal discussion, John, you mentioned about PhDs with a more commercial focus, an element of commercial focus of modules. I mean, is that one small way that could help? What's your point of view on that, John?
Yeah, so there's more and more of that happening. So there are ways of gaining PhD credits where you spend some time doing normally a mock entrepreneurial activity based on the research in one's PhD. You can do this as a postdoc as well at the early stages or even later, and they can be quite useful. But quite often they don't really go into the nitty-gritty of things. And I think what might be better is to have actual placements within PhDs that are certainly within companies. That's been happening for years anyway, but something more in the tech transfer side of things, or certainly between manufacturing and basic research in gene and cell therapies. I think more crossover actually as part of the real day-to-day work would be useful, as well as the training exercises that people are already doing.
Yeah, I think that will certainly give that foundation. I think some of the advice from a recruitment perspective we're giving companies now on a regular basis is looking for those who can transfer their skills in. And a lot of companies now are giving those who are at early stages of their careers the platform to get exposure in different areas of the business. So not just say stuck at the bench, what is project management, what is even business development, or different functions within the business. The idea is then to give that more rounded education to help the career develop further forward. And certainly I know that's very attractive to candidates. Doesn't matter whether you're at the start of your career or you're heading towards a director or a CSO level, that exposure to help with decision making in the business environment is definitely very, very important. And but staying on that theme of challenges, we talk about them a lot, right? And sometimes they're come at very difficult times during that scale up process. But to overcome challenges, you need that right mindset. So realistically, I think it's quite a simple question this time around. We start with you, Kerstin. What is the right mindset to scale up a cell gene therapy? What would you say the key attributes needed are?
Well, we've talked about many of those already, right? And they don't differ that much from like entrepreneurial mindset generally. So you have to have like this kind of pioneering attitude. You have to be resilient. We have mentioned the point about teamwork and being open to have discussions across silos. You have to be able to speak different languages, right? You need to be able to converse equally well with your investors as with your scientists, as with the regulators. So that's actually quite a lot of languages. And then most of the companies that I manage, they have a healthy mix of computation in there, which is a different bucket of languages altogether. Yeah, and then generally, I think a good dose of humor always helps.
Yeah, I agree. Being able to have a bit of a laugh and a sense of humor is very important. But yeah, languages, well, as you say, different countries, but also, when you start talking computationally, scientifically, it's very hard to switch from one to the other. So being more well versed in what people bring will definitely help. But John, what about your point of view in terms of that right mindset from, because your perspective being slightly different, but be very valuable?
Certainly perseverance in biotech is very necessary. You don't get instant reward always, and it's a bit of a tricky one to sometimes navigate. Sometimes things that should work on paper don't work immediately. That can be very difficult for people at early stages, I would say. So perseverance is a massive attribute that's required. Certainly as well, what I've found, even getting to the point where I have to start thinking about how am I going to scale up this therapy, or how are we going to get it to clinic, a lot of the time there's been no pathway at all. Even on the regulatory side, people aren't really quite sure what's going to be required, and I have to come up with suggestions, or I have to come up with a pathway, and sometimes decide out of all of the experts that have been consulting who to listen to and who to ignore, and to make my own decisions. And that can be quite difficult when you're used to having somebody always to ask that can tell you what the right pathway is. You've got to be creative and sometimes headstrong enough to figure out what is the right way to do it, which is exciting, but also can be slightly daunting at certain points. And yeah, just a bit shocking for me when I realized that's kind of how the space is. But yeah, opportunities there, definitely.
Yeah, I think your word's shocking there. That I've never that describes it perfectly, because it's got me right, and I haven't really even been involved in it, and never really heard of that point of view. When you're in your position, having to make decisions without that pathway, and yeah, headstrong most definitely, but also that perseverance and just resilience. I mean, to be able to come to a conclusion and then put it forward, I'm impressed. That's what I can say, definitely. But Stephen, from your point of view, in terms of how you see that mindset in those you speak with, and what do you see as the key attributes?
I think three points. One would be ambiguity tolerance, which, if you're an early developer in today's disrupted world is a must-have. We all have to deal with disruption and new situations evolving. So that's an important thing, and realizing that there's many different ways to learn and through experience. So I think that that point number two would be humility. So realizing where your area of expertise ends and not extrapolating beyond that. So for example, if I'm an early developer, I've grown up in a world that is focused on publication, published on novelty, ideation. The next silo on, is very different, and things like consistency and producing a product that hits specific specifications, understanding variability in your process, very different cultures. So what Kerstin was alluding to earlier in terms of like being able to speak different languages, you've also very different cultures. So people who are very good openers, early developers tend to be great openers, but then later on you need closers, people to whittle things down and to really focus and push stuff over the line. So I think what comes back to the cross-functional teams. Having to be a good team player, you have to have a degree of humility and know where your expertise no longer pushes the team effort forward. And that that takes a bit of humility and self-awareness. And then the third thing, I think, is realizing that it takes time, and that you won't be able to do it all by yourself. So and that it will take time. So I think finding out where you're strong and where you're weak, not being afraid to try things just for the experience, because the experience can yield a lot of information that you won't find in textbooks. I think that those would be the three I would list on top of what Kerstin and John said.
Yeah, no, I think definitely the humility got me there, because I think you're quite right. A lot of the time we often can find ourselves pretending to talk about what we don't really know, but of course the impact it must have in that environment when you're trying to, there's so many different contexts. Yeah, humility is a good point well raised, often not one that I've often hear. More often it's perseverance and ambiguity. So yeah, thank you, appreciate that.
Yeah, I think it's pretty hard, you know, when you come out of a hyper competitive environment of academia to actually be vulnerable enough to actually question yourself where I'm deficient. And, you know, I think business people, a lot of the time, they're completely focused on development of teams because they realize that you know, the quality of the team governs the quality of the outcome.
Oh,
That's
so
a
the
really
difficult
good point. Sorry, John, you go.
Sorry, did you go first?
Oh, I was just there was a really good point that Stephen just made, and for our entrepreneurs that we want to work with and start companies with, testing for empathy is one of the criteria that we pay a lot of attention to, and it's quite peculiar that traditional academic path actually drives you away from that. And that's the most common indicator where people tend to score low if they join us straight from academia. So we're actually testing for ways whether this is real or whether this is just an acquired behavior that the academic pathway has instilled. So this is quite interesting, actually.
Yeah, that is really interesting. That's really interesting. John, you were going to say something as well.
Yeah, I would just say maybe kind of challenging the point slightly. I think certainly when you're trying to start something and when you're trying to lead something on a journey and trying to get people behind you, try to push something forward, sometimes you do have to be able to understand everything because you've got to lead every angle. You can't just say, okay, I'll trust that person with that information. I'll do whatever they say. So to a certain extent, you do almost have to systematically reinvent yourself to be able to understand each aspect to a certain degree. Obviously, as you say, having the humility to know the limit. But I think it's a slightly different situation when you're at the inception of something to when it's already established. And so you do have to be like a one-man band to a certain extent until you get to that point where you've got sufficient backing. And that probably then does stick in people's minds when they've been at that stage and then kind of just have to let go of certain things. So I think that's possibly where it comes from as well.
Yeah, I mean, like in general business circles, the kind of people you want to set up a business are very different from the kind of people you want to scale the business. And it's exactly the same in cell and gene therapy development. So, you know, you do require the grit to go it alone, but then at the right stage, you need to choose your friends and form a team.
Yeah, I think it's difficult, isn't it? Because sometimes in business you have to make business-based decisions, which are more on facts and figures rather than sort of the more personal side of things, and that can be quite difficult. You need to find that relationship where you can actually talk in that open manner and make those decisions in the right way. So very crucial. But conscious of the time, we do have, and I could keep talking on these points for ages, but I'm sure we all could. But got a couple of questions. So this one's for yourself, John. You obviously going back earlier in the conversation about different strategies for vector introduction in the development of products where cross-species genetics, so for example, by use of recombinant DNA, have been in play. Do you have a preferred method, or which method would be best in that context?
I could probably say the one that I'm trying to commercialize. That would be a bit naughty, really, wouldn't it? But I'd say it kind of totally depends on what the aim is with the intervention. For example, if the aim is to, in the case of spinal muscular atrophy, they've used an adeno-associated virus vector to deliver the gene into the right cell types in the body, and they're doing in vivo gene therapy there. Quite often, the reason that AAV is used in an in vivo setting is because quite often it doesn't need lots of integration for stable expression when it's delivered in vivo, and it's got really good biodistribution, gets into a lot of cell types, and can be customized as to what you need from it. But for some of the ex vivo gene therapies, if you're looking to deliver DNA into dividing cells like stem cells or iPS cells or CAR T cells, whatever, where you're going to amplify those cells, generally you need something that's going to integrate into the genome, and then people use a retroviral vector, which comparatively wouldn't be any good or normally that good for doing in vivo gene therapy, depending on what it is you're trying to do. So it really starts with what the intervention aims to do. And Yeah, I'd say that the two leading DNA delivery systems are AAV and lentivirus. There's others coming in, but I'd still say that if I was starting a project now, or if somebody said, I want to do a serious therapy, get this to clinic, want it to be something that's scalable, can be commercialized, those two have definitely got the processes there. And they've already been in the clinic in different contexts as well. So one of those, depending on whether you do an in vivo or ex vivo. Or in my case, I'm kind of mixing up a bit, so I shouldn't say that. You can use them for anything.
Amazing. Thank you. Appreciate that. I mean, Kerstin, I mean, on that same question, do you have a point of view or perspective?
Oh, I can only repeat myself. Horses for courses. Really depends on the indication.
Yeah.
I mean, there's obviously different considerations, not just what works, but also what's novel, what puts you in a really good IP position, right? So all these factors weigh into that decision that you have.
Yeah, no, I suppose it's true. We sort of kind of cover the same ground again. And I mean, Steve, you feel the same, or is there anything else you'd like to add?
Yeah, I think, people tend to tribalize around technologies and get very fervent over like their particular type. And, I was reading a very good book recently of Flesh Made New, which discusses kind of huge problems that have happened historically in the regenerative medicine space. And part of that is where dogma is adopted over, data. That I've done 17 years using this kind of technology. The new technology, it's not half as good because I'm invested in the older technology, or vice versa. And that we really, all of us always have to be led by the data rather than, I think how much work we've put into a particular technology.
I see. Because that could be, I must admit, I can see how that could be quite a challenge getting stuck in, just because you say you've been working this for 17 years, that must be the only way. So, yeah, so preparedness to always be open to new ideas, new concepts with data behind them. There's a new avenue, a new way to follow, to develop. I think, yeah, sometimes it's easier said than done, most definitely.
I think one of the key things as well is just because the technology is new doesn't necessarily mean it's the best technology to introduce into your therapeutic development program. So, for example, with iPS cells, at the moment, the vast majority of people I deal with, they're using Sendai virus that has been around for donkey's years. But it's very well known to the regulator. The risks associated with its use clinically is very well measured. And, there's many clinical trials that have been using it. So it's seen by many people as the quickest route and to getting regulatory approval for kind of therapeutic development. So, this is what I was talking back earlier about, like cross-functional teams and having the discussion with people in different silos so that you make life as easy for the therapeutic to be brought to the clinic as quickly as possible, while maintaining obviously safety and the efficacious nature of the therapeutic.
Yeah, well, most definitely. And thanks for that. I think also, could you mention the name of that book once again? Is just
Of Flesh Made New. So it's written by John Roscoe, who's ex-president of ISCT, the International Society for Cell and Gene Therapy. So John Roscoe of Flesh Made New. It's a brilliant read. I read it in one sitting, and you're there, and that it's all talking about the importance of always focusing on the clinical reproducibility of what you're trying to make. And we can get sidelined by other things, so it's important to keep our eye on the prize.
Most definitely, yes. And just keeping on time, because we could keep going for a very long time. But so let's try and squeeze in one more very, very quickly. I guess, so question, it's about the readiness of translating CGT compared to other major advanced economies. So I was pausing, that's such a big question. So, I mean, how's the UK's readiness in translating CGT compared to other major advanced economies? I guess starting, well, Kerstin, I mean, what's your point of view on that particular question? Just sort of very quickly.
I think overall it's pretty good, right? So obviously I think the US and Japan, they are some of the kind of leaders in terms of what's happening there, in terms of progressing things into the clinic and also from a regs perspective. In Europe, I think the UK is one of the leaders. I still think that Brexit has been a massive problem in terms of losing talent, and a lot of people are trying to sell it to me as an advantage because we can make up our own rules and whatnot in terms of regulation, but I still don't see it that way. If I'm a biotech company and I want to have my product exposed to the widest market possible, then I will still choose the widest market. So I'd say overall pretty good. But yeah, we need to really understand that we are competing globally. So there's a lot to be done in terms of access to capital, I think, and making sure that the talent pool is being replenished and doesn't run off.
Yeah, no, I think I agree with you about the talent pool. It's been challenging, but don't want to get too bogged down in that or distracted. But John, what's your point of view in terms of UK's readiness from what you've seen?
The UK's readiness for what exactly are we focusing on here?
So it's UK's readiness in translating CGT, so therapies compared to other major advanced economies.
Well, I guess there's been a long history of CGT in the UK. It seems as though we're very competitive in this field compared to some of the other economies. Certainly when I go around to conferences internationally, there's always a big showing of the UK groups. So I'd say, yeah, it's pretty strong as far as I can tell, but I'm really coming from a very early development stage, where I'm probably going to be seeing a lot of the research groups rather than the companies and the finished product.
Yeah. No, thank you. I mean, I know you're at various stages, you're still very, very valued. And Stephen, of course, I know you've got.
Okay, I've got a positive spin on Brexit. In that, the government realizes that there's an economic downside to cutting yourself off, but I think they're seeing cell and gene therapy as one of the areas that's going to be strategically important in minimizing that economic damage. So I think that actually cell and gene therapy will be more enabled because of the damage caused by Brexit to the economy overall. So I think they're seeing it as a strategically important area, given the UK's strong scientific expertise, its strong regulatory environment. And I think the infrastructures, as I alluded to earlier, are, they're fused together. So, infrastructures like the catapult, the NHS, very hard to build without a long-term vision. And there's been some very smart people in the UK that have thought long-term. And, if we deal with the other bottlenecks, like, for example, enabling early developers to get access to the cross-functional teams to make the right decisions, the consistent funding of early stage development through to a company becoming self-sufficient and be able to generate its own revenue. But I think that, the cell and gene therapy space isn't going away. And I think the UK is kind of doubled down on the space because of the Brexit decision in particular.
Yeah, no, it's well, certainly yes, the cell and gene therapy space is not going away. It's only going to grow and accelerate with that growth. But thanks for that positive spin on Brexit. I think there's always going to be sort of polar opinions on it, which are good to hear, most definitely. And I think from my perspective, I could keep asking more questions, but unfortunately we've run out of time. But I've got to say, John, Kerstin, Stephen, thank you so much for your time and input. Really appreciated it. And yeah, I've certainly learned a lot, and I hope everyone else listening and joining us has. And also those in the audience, just thank you for taking the time to spend your morning, evening or lunchtime with us. And once again, speakers sharing your insights. Of course, there will be more webinars, particularly on this topic of the cell and gene therapy sector, and led by my colleagues as well in the coming months. So please, they'll be announced in the next coming weeks. Please watch out for them and join us. But final point, for those who wish to continue the discussion further, please do join our networking room next. Use a link in the chat to meet our panel, dive deeper, ask questions on the topics we discovered today. But that's it from myself. Thank you everyone for attending. Thank you, the panel, once again. Really appreciate your time. And yeah, thank you. And hopefully speak again very soon.